Hey everyone, this is Dr. M with another supplemental video for my genetics lecture course. This video is going to discuss when exactly during meiosis we go from having 2 copies of chromosomes to having 1 copy of chromosomes. So just to review, chromosomes are comprised, each chromosome is a single DNA molecule, unless the chromosome has replicated, in which case it then is 2 DNA molecules that are attached to the centromere. These are 2 identical sister chromatids. Keep in mind that its homologous sister chromosome, its homologous chromosome is genetically similar, but not necessarily genetically identical, because the sister homologue may contain different alleles. However, these 2 sister chromatids are genetically identical because they are exact copies, in theory, of each other. Here is what one of those looks like. Again, there are 2 DNA molecules here because this chromosome has replicated, but still it is 1 single chromosome. So in the normal cell cycle during mitosis, we don't ever say that at the S phase, when chromosomes replicate, we don't suddenly double our number of chromosomes when we replicate them. We still have the same number of chromosomes. So what this looks like during metaphase of mitosis is that chromosomes line up on the metaphase plate somewhat according to this. We can envision them 2-dimensionally, well, rather 1-dimensionally really, in a straight line on the metaphase plate. Now here we have 2 sets, 2 copies of 2 chromosomes, and our 1 copy is in blue and 1 copy is in red. 1 chromosome is big, so we have a big red, a big blue, a small blue, and a small red, okay? They're lining up on the metaphase plate such that when anaphase happens, which is here, each of the sister chromatids get pulled apart and each of the daughter cells gets its own complete set of sister chromatids. The chromosome number throughout this process stays the same, okay? So here we have 4 chromosomes. Here we have 8 chromosomes, but each of the daughter cells after division gets 4 chromosomes, okay? All right, so now in meiosis, what happens is that we have 2 cell divisions. We have meiosis 1 and meiosis 2. In the first cell division, that's when chromosome reduction happens. And so after the first cell division, we start out with 2 pairs of 2 copies of chromosomes. So we have 4 total. We have, again, as in the last slide, 2 large and 2 small, designated by color. And after this first cell division, each cell goes from having 4 chromosomes total to having 2 chromosomes total. They get one of the homologous chromosomes, okay, and the other cell gets the other homologous chromosomes. The second meiotic division, meiosis 2, is very much like mitosis, such that in this division, if you just focus on the cell up here, these 2 daughter cells are genetically identical to this one. And here, these 2 daughter cells are genetically identical. Well, barring crossing over, but let's just forget about that for a second. Okay, and so what this looks like in metaphase 1 is that rather than cells lining up all in a line, which is what we saw in mitosis, the sister homologues, the homologous chromosomes, are pairing up side by side. And so each cell, each of the daughter cells, is going to end up taking an entire sister chromosome with it. And so the sister chromatids are still connected at this point. So I just want to emphasize that here we have 4 chromosomes. We start out with 4 chromosomes, and now each of our cells has 2. So here's where the reduction in chromosome number happens. For the second metaphase, now we line up like we would expect in mitosis. And so essentially now here is showing crossing over. And so ultimately, the daughter cells are not necessarily exactly identical to the parent cell or to each other, but in terms of how the division happens, how the segregation of chromosomes happens, it is just like mitosis, such that sister chromatids are separating from each other here at this point. Okay, so just to review, this is the mitotic, this is in mitosis, this is the mitotic metaphase plate. And here sister chromatids are going to be separating. So that's it.